So this be a 3-cell fascicle right here. And so the pink is the sarcolemma, the actual cell membrane. All right, and the gray would be the endomysium connective tissue surrounding each individual cell. The blue dots would be the nuclei of the connective tissue cells. They've got the gray in between individual muscle cells, the gray in between the cells. And here, capillaries run through the endomysium. So there'd be endomysium here, the capillary running through it, and so on. All right, longitudinal view reveals more than one nucleus. This is one skeletal muscle cell showing a nucleus here, nucleus there. These two bulges, more nuclei. So look at a cross-sectional view. Very superficial position for the nuclei, just under the sarcolemma, just immediately deep to the cell membrane. Okay, let's see. If I could, anybody happen, let's see, borrow your pen for a second, see if this will work. Thank you. And here you're recording me vandalizing a model. So I could get in a whole lot of trouble here. See if this will mark in. Nah, not quite doing what I wanted to do. Okay, that's dark enough. We can kind of see it. Thank you. And you see what I've darkened there? The section I've darkened, the little outline around, that's one myofibril. So within one myofiber, within one muscle cell, are many myofibrils. I actually inked in one myofibril. And you see little dots in the center of that myofibril? Those dots are myofilaments, thick and thin myofilaments, although this model makes no attempt to differentiate between thick and thin myofilaments. It's just generic myofilaments in here. Okay, so can you tell me what's wrong with that cross-sectional view? They screwed up. Tell me how they screwed up. Do you have any problem with that nucleus there? Yes or no? Do you have a bone to pick with that nucleus right there?
Yeah, so this is supposed to be deeper.
No, it's skeletal muscle cell. Very, very shallow nuclei. So is there any problem with that nucleus here? No. Which nuclei have the problem? Those two. They're too deep. They're too far in. Yeah. So you do not see nuclei in anywhere near that far.
So why did they put it there?
Because they screwed up. They made a mistake. They made two dumb mistakes. So remember, this is correct. Just under the sarcolemma. These two very shallow nuclei. They're correct. Okay. What else can we point to on here? Striations. Longitudinal view. Every red band perpendicular to the long axis is a striation on here. Let's see. Oh, and this. Here's an axon collateral, just like the left side board up there. An axon collateral of one somatic motor neuron leading to the motor end plate. So this brown patch of the sarcolemma is the motor end plate. So axon collateral leading to the motor end plate of this one skeletal muscle cell. So you've got all these synaptic end bulbs packed with vesicles filled with acetylcholine. And so the motor end plate has the nicotinic cholinergic receptors for the acetylcholine. The rest of the sarcolemma does not have nicotinic cholinergic receptors. There's only a small patch of the sarcolemma of a muscle cell with the nicotinic cholinergic receptors. The rest of the sarcolemma has voltage-gated ion channels for the generation and propagation of an action potential. So you could get the acetylcholine binding the nicotinic cholinergic receptors. You would get depolarizing graded potentials that would spread out. They'd reach these voltage-gated channels. They would open up, and you would get an action potential traveling in two directions at once, away from the motor end plate. Okay, that's it for this model. Any questions for me?